When we picture a medical emergency, an ambulance with sirens blaring or doctors rushing into a trauma bay, we always think of it at the human scale. We see the big picture like a heart stops beating or a lung stops breathing.
Right. Yeah, but that is really just the macro view. I mean, the real drama, the actual literal life or death struggle is happening on a scale so profoundly small that you can't even see it without a microscope.
Exactly. And that's exactly what we are getting into today. Because every single medical emergency you can think of, whether it's a massive heart attack, a stroke, frostbite, or even a Tylenol overdose, it's fundamentally a race against a microscopic ticking clock inside your cells.
Yeah, and today we've gathered a really fascinating stack of sources. We're looking at medical pathology textbooks, foundational biochemical research papers, and modern clinical emergency guidelines.
Our mission in this deep dive is to unpack exactly what happens when our cells are pushed past their absolute physical limits. We are exploring the medical pathophysiology of cell injury and cell death.
Which I know sounds a bit morbid right out of the gate, but it is actually the absolute bedrock foundation of modern medicine. It's about understanding the exact, precise molecular threshold between life and death. Because figuring out how to manipulate that specific threshold is how we actually manage to save lives in a hospital.
Okay, let's unpack this. Before a cell is pushed to the brink of death, what does it actually need just to survive on a, totally normal, quiet Tuesday afternoon?
Well, to understand that, you kind of have to think of a healthy human cell as this incredibly complex, highly certified city, but one that takes a massive, continuous amount of energy just to maintain its basic walls.
Right.
In pathology, we look at 4 core pillars of cellular survival. And if any of these 4 pillars crumble, well, the cell is in immediate trouble.
So what is pillar number 1 then?
First, ion gradients. The cell has to meticulously maintain a steep chemical gradient across its outer boundary. It has to keep high levels of potassium inside the cell and pump high levels of sodium and calcium outside.
It's basically actively pumping ions against their natural flow, right? Like constantly pumping water up a hill.
Precisely. And pumping water up a hill takes energy, which brings us to pillar number 2, a continuous, uninterrupted supply of ATP.
The energy currency.
Yeah, the universal energy currency, primarily derived from oxygen. Then pillar 3 is perfectly intact cell membranes, so the walls of the city. And pillar 4 is correctly folded proteins, the tiny machines that do all the actual work.
Okay, so let's play this out. Let's say the oxygen supply gets cut off, like a blood clot blocks an artery and blood flow just stops. What happens to those 4 pillars?
Within a matter of seconds, the cell's main power plant starts to slow down. This is a process called oxidative phosphorylation inside the mitochondria. ATP production just absolutely plummets.
Wow.
And here is where the immediate cascading crisis begins. Remember that first pillar, the ion gradient?
Pumping the water up the hill?
Right. Maintaining that gradient requires a staggering amount of energy. There is a specific molecular machine called the sodium-potassium pump on the cell membrane, and it uses roughly 20 to 40% of a resting cell's entire energy budget.
Wait, 20 to 40% of its energy just on maintaining that one single balance?
Yeah, it is that vital. It just keeps sodium out and potassium in. So if ATP production halts because of a lack of oxygen, that pump runs out of fuel and fails almost instantly.
Oh, I see.
And when the pump fails, all that sodium that was being kept outside the walls comes rushing right back into the cell. And in biology, there is a golden rule. Where sodium goes, water follows.
Because of osmosis, right? The water rushes in to dilute the sodium.
Exactly. So the cell and all its tiny internal organs just start physically ballooning with water.
Like a water balloon.
Yeah, microscopically under a slide. Pathologists call this hydropic change. The cell is swelling up. And at the exact same time, because the main power plant is down, the cell desperately switches to a backup power system called anaerobic glycolysis.
Anaerobic, meaning without oxygen, kind of like when you're sprinting really hard and your muscles start to burn?
Yes, exactly like that. It generates a tiny bit of ATP to keep things alive, but the exhaust byproduct of that backup system is lactic acid. So as lactic acid builds up, the internal pH of the cell drops, and it becomes highly acidic.
It sounds bad.
Very bad. That acidity causes the DNA, the chromatin inside the nucleus, to physically clump up, which shuts down the cell's ability to make new proteins.
I always picture this early stage of injury like a leaky boat out on the ocean. The cell is the boat, and that sodium-potassium pump is the bailing pump, constantly churning and spitting water out just to keep the boat afloat.
That is a great way to visualize it.
Right. And if the engine dies, meaning we lose our oxygen and our ATP, the bailing pump stops working, water starts pouring over the sides, the boat fills up, and it swells. But, and this is a really crucial point for you listening because the sources emphasize this heavily, at this stage, the boat hasn't sunk.
Right.
This injury is entirely 100% reversible.
That is the key clinical takeaway right there. If you restore blood flow, if you restart that cellular engine, the bailing pump turns back on.
It just clears the water out.
Exactly. It rapidly clears the water out, the pH normalizes, the DNA unclumps, and within hours, the cell looks structurally totally fine. It survives as if nothing ever happened.
Okay, so the leaky boat can be bailed out, provided you get there in time. But if the cell can fix itself, what is the exact mechanism that pushes that boat underwater for good? Like, when does reversible injury cross the line and become irreversible death?
What's fascinating here is that there is a highly specific, identifiable biochemical threshold. It isn't just this gradual fading away. The point of no return is all about one specific element, and that is calcium.
Calcium? Like, what's in my bones and my glass of milk?
Yes, but inside a cell, free-floating calcium is actually highly toxic. Remember how we said calcium is supposed to be kept strictly on the outside of the cell?
Right, yeah, that was part of pillar number 1. Keep the sodium and the calcium out.
Exactly. So as the cell swells and its membrane integrity starts to physically stretch and fail, you get this massive, sustained influx of calcium rushing into the cell.
Oh, wow.
And specifically, it rushes into the mitochondria, which are the cell's power plants. This massive calcium overload physically forces open a microscopic structure called the mitochondrial permeability transition pore, or the MPTP.
The MPTP, so it literally punches a hole in the power plant.
That's exactly it. And once the MPTP opens, the mitochondrion loses its internal pressure gradient entirely. It can never, ever make ATP again.
The engine is dead.
The engine is dead, and the blueprints to rebuild the engine are destroyed. But it's actually much worse than just a permanent power outage.
How could it be worse?
Well, that flood of calcium acts like a master alarm, triggering a cellular self-destruct sequence. It actively turns on a whole set of highly destructive enzymes inside the cell.
Okay, wait, you just mentioned a bunch of destructive enzymes. I mean, the sources list things like phospholipases, proteases. Let's slow down and break those down. What are these enzymes actually doing to the cell?
They are systematically dismantling the city. Phospholipases specifically attack lipids, meaning they literally dissolve the cell's fat-based outer membranes.
They melt the hull of your boat.
Basically, yes. And then proteases degrade the structural scaffolding proteins, so the cell loses its physical shape.
Yikes.
Yeah. Endonucleases act like molecular scissors. They enter the nucleus and shred the cell's DNA into tiny, unreadable pieces. And finally, adipases wake up and aggressively burn through whatever tiny microscopic bit of backup energy is actually left.
So the calcium comes in and basically hands weapons to the cell's own proteins to tear the place apart.
Exactly.
And eventually, the outer wall breaks entirely, these enzymes leak out, or acid-filled sacs called lysosomes leak in, and the cell undergoes autolysis. It just digests itself from the inside out.
Yeah, once that pore opens, even if you restored 1,000 liters of oxygen, it is too late.
It is entirely too late. But wait, let me push back on this a bit. Is this a universal timer? Like, if oxygen gets cut off in my body right now, does every single cell from my brain to my toes hit this calcium threshold at the exact same minute?
Oh, not at all. And that is a critical nuance in emergency medicine. The threshold varies drastically depending on the cell's specialized job and its metabolic demands.
So some are faster than others.
Very much so. Neurons in your brain are exquisitely sensitive to oxygen loss. They are total energy hogs. They can suffer irreversible injury, meaning they cross that calcium point of no return in just 3 to 5 minutes without oxygen.
Whoa. Just 3 to 5 minutes and the brain cells are gone.
Exactly. But your skeletal muscles, the cells in your legs or arms,
Right.
or the connective tissue in your joints, they can tolerate much, much longer periods of ischemia, sometimes hours, before hitting that irreversible threshold.
That is a huge difference.
It really is. That incredible variability is exactly why the clinical criteria for declaring brain death versus cardiac death are fundamentally different.
Oh, that makes sense.
Yeah, it's also why organ transplant protocols have very different acceptable time windows. You can keep a kidney or a cornea on ice for vastly different periods than you can a human heart.
Okay, so if a lack of oxygen is the original trigger that starts this whole leaky boat, calcium flooding disaster, the obvious medical solution seems simple, right? Just give the patient oxygen as fast as humanly possible.
Right, you'd think so.
Yeah, just open up the blocked artery and flood the tissue with blood. But in reading through our pathology text today, there is a pretty terrifying catch to that logic.
It is absolutely one of the most counterintuitive phenomena in all of human biology. It is a condition called reperfusion injury.
Reperfusion injury?
Yes. You would logically think that restoring blood flow and oxygen to a dying, suffocating tissue is always the ultimate life-saving move. But if you restore a massive wave of oxygen after a cell is already calcium overloaded, but before it has fully, completely dissolved, you can actually accelerate its death.
Wait, stop. That makes zero sense.
I know, it sounds crazy.
We literally just spent 10 minutes establishing that the cell is dying because it lacks oxygen to make ATP. How can giving it its fuel source back become the very thing that kills it?
Because you are introducing volatile fuel to a shattered engine. The cell's mitochondria are already severely physically damaged and flooded with calcium, right? They are broken.
Right, the pore is open.
Exactly. So when you suddenly introduce a massive rush of pure oxygen to that broken machinery, it doesn't calmly make ATP. Instead, the process misfires.
How so?
It undergoes incomplete reduction, generating a massive burst of reactive oxygen species, which we commonly call free radicals.
Free radicals, so oxygen with unpaired electrons that just want to rip through everything.
Exactly. They act like microscopic shrapnel. The sudden burst of free radicals rapidly destroys cell membranes and finishes off cells that might have actually been salvageable.
If the oxygen had just been reintroduced more carefully.
Right, or if the free radicals had been neutralized.
So the rescue mission ends up blowing up the survivors.
That is exactly what happens. And this isn't just theoretical pathology. There were classic foundational experiments done in the 1970s and 80s by researchers named Jennings and Reimer.
Okay, what did they do?
They were studying dogs to see exactly what happens during a myocardial infarction, heart attack. And they discovered that the muscle of the heart doesn't just all die at once when an artery gets blocked.
It doesn't.
No, it dies in a highly predictable spreading pattern.
Yeah.
Kind of like a wavefront on a beach.
A wavefront, so it starts in one specific geographic place in the heart and moves outward.
Right. Think about the anatomy of the heart wall. When a major coronary artery on the outside of the heart is totally blocked, the cells furthest away from that blood supply hit that irreversible calcium threshold first.
The ones on the inside.
Yeah, right under the inner lining of the heart chamber, an area called the subendocardium. They are at the very end of the dried up water pipe.
So they die first.
They usually die within 20 to 40 minutes. But as the blockage continues, this zone of death spreads outward toward the outer heart wall, the epicardium. Over the course of about 3 to 6 hours, that wavefront of death sweeps through the entire affected territory.
Which completely explains the mantra you always hear in emergency rooms, time is muscle.
Exactly.
Or why cardiologists constantly track door-to-balloon time. They're literally racing to get a tiny balloon into the blocked artery to open it up before that wavefront of irreversible calcium-driven death sweeps through the entire thickness of the heart wall.
And, crucially, they have to manage the return of that blood and oxygen so they don't cause massive reperfusion injury with those free radicals.
Right, the shrapnel.
Yeah. That's why emergency cardiology and stroke medicine for the brain have built entire clinical protocols around this. Things like controlled reperfusion, therapeutic hypothermia to slow the metabolism down, and staged reoxygenation, all to manage this exact paradox.
Hearing about free radicals destroying cells really connects to something I remember from the 80s and 90s.
Oh, yeah.
Yeah, there was this massive, inescapable antioxidant supplement craze. It was on, every vitamin bottle in the grocery store.
Oh, absolutely. The marketing was brilliant and completely ubiquitous.
Right. And it came from a real place in the literature. A scientist named Denham Harmon formalized the free radical theory of aging way back in 1956.
Yes, a very famous paper.
It was real peer-reviewed science about oxidative cell injury, but marketers just ran with it. They oversold it to the public, claiming high-dose antioxidant pills could neutralize all free radicals, stop aging, and prevent every disease.
Which is a huge leap.
Exactly, because when researchers actually ran large, rigorously randomized clinical trials testing things like high-dose vitamin E and beta-carotene, they found absolutely no benefit, and in some cases, it actually caused harm.
Right. If we connect this to the bigger picture, it's a perfect historical example of how a very real, incredibly nuanced cellular mechanism like oxidative stress gets completely flattened when it's translated into a consumer product.
You can't just swallow a pill for it.
You simply cannot just swallow a handful of vitamins and expect it to reach a localized biochemical wavefront of death in a blocked heart artery.
Okay, so the wavefront sweeps through, the cells burst from the free radicals. But wait, if all these cells are bursting open and spilling their broken engines and toxic enzymes everywhere, what does the tissue around them do? Doesn't that cause massive inflammation?
It absolutely does, and that brings us to a major pivot in how we understand pathology. For a long time, medical science viewed cell death as a single passive endpoint.
It's just one type of death.
Right. In 1858, Rudolf Virchow, a brilliant German physician, coined the famous maxim omnis cellula et cellula. Every cell arises from a cell.
He essentially founded the entire field of modern cellular pathology.
He did. But back in his day, they thought of cell death just like a rusty machine breaking down. Pure, passive, inevitable decay.
But that model had a huge flaw, right? I mean, it couldn't explain why the wavefront of a heart attack caused violently messy swelling and massive immune reactions, while other cells in the body seem to just, quietly disappear during normal human development with absolutely no fuss or inflammation at all.
Precisely. Because they were actually observing two completely different biological categories of death, what we now call necrosis versus apoptosis.
Okay, let's define those.
Necrosis is the messy, accidental death. It's the leaky boat sinking violently. The cell is overwhelmed by damage like a lack of oxygen or a toxic chemical. It swells, the membrane ruptures, and all its internal contents spill out into the surrounding living tissue.
Just everywhere.
Yeah. That toxic spillage acts as a massive alarm bell, triggering heavy inflammation and drawing in white blood cells to clean up the mess.
So necrosis is the chaotic, violent death, but apoptosis is the tidy one.
Very tidy and highly intentional. Apoptosis is an active process. It actually requires the cell to spend ATP to do it.
Oh, so it uses energy to die.
Exactly. It is a meticulously genetically encoded program the cell executes on itself. Instead of swelling and bursting, the cell shrinks. Its DNA is cleanly and deliberately chopped up into specific lengths, and the cell breaks itself apart into neat, membrane-bound little packages.
And then what happens to the packages?
These packages are then quietly eaten by neighboring cells. There is no spillage, no alarm bell, and zero inflammation.
Here's where it gets really interesting. The origin of the word itself. In 1972, researchers Kerr, Wiley, and Curry published a landmark paper formally describing this tidy, programmed death.
Yes, a very famous paper.
Right, and they needed a good name for it. So they consulted a Greek scholar and chose apoptosis, which is actually borrowed from an ancient Greek word used to describe leaves falling from a tree in autumn.
I've always loved that.
Me too. I just love that. It's the absolute perfect metaphor for a natural, non-destructive, seasonal loss of cells.
It is a profoundly beautiful biological process. But as a clinical pathologist, I have to say that even the messy death, necrosis, has a vital diagnostic utility to it.
Really? How so?
Because the pattern of that messy death acts as a literal fingerprint for the specific disease that caused it.
Give me an example of that. How can you tell what killed the cell just by looking at the mess it left behind under a microscope?
Let's take tuberculosis, a bacterial infection in the lungs. When the immune system encounters TB, it creates a prolonged standoff that results in a very specific type of tissue death called caseous necrosis.
Caseous meaning cheese-like.
Literally, yes. It leaves this soft, crumbly, yellow-white, formless center in the tissue, destroying both the cells and the lung architecture.
Wow.
Pathologists can look at that highly specific, cheesy mix of dead cells, mycobacterial fat, and immune debris, and often confidently name the underlying disease right on the spot.
And there was another crazy example in the sources, one that happens all the time in emergency rooms, acetaminophen overdose, so Tylenol.
Yes, this is a classic example of toxic cell injury. At normal, safe doses, your liver easily metabolizes Tylenol. It produces a tiny amount of a highly toxic byproduct called NAPQI. But your liver neutralizes it instantly using a natural cellular sponge called glutathione.
Okay, so glutathione is the sponge. But in a massive overdose...
In an overdose, you completely deplete all your glutathione. The sponge is fully saturated. So that toxic NAPQI accumulates rapidly and begins directly binding to liver proteins, destroying them.
Ouch.
But here's the fascinating part. It doesn't destroy the whole liver randomly. The liver is divided into microscopic anatomical zones. This overdose causes highly specific necrosis only in zone 3 of those liver lobules.
Wait, why only zone 3? Why not zone 1 or 2?
Because that specific anatomic zone, which sits right around the central vein draining the liver, happens to naturally have the highest concentration of an enzyme called CYP2E1.
Okay.
And that is the exact enzyme that generates the toxic NAPQI in the first place. It is a textbook anatomical fingerprint, the cells that make the poison die first.
That makes perfect sense.
And understanding that specific biochemical mechanism is exactly why we know how to treat it. The universal antidote for a Tylenol overdose in the ER is a drug called N-acetylcysteine.
Because it builds more sponges.
Exactly. It works simply by giving the liver the raw amino acid materials it needs to rapidly build more of that glutathione sponge.
It all connects back to understanding the fundamental cellular mechanism. If you know how the boat sinks, you know how to patch it.
Exactly.
But going back to the tidy death, apoptosis, the discovery of how perfectly programmed this is actually won a Nobel Prize, right?
Yes, the Nobel Prize in Medicine in 2002. Sidney Brenner, Robert Horvitz, and John Sulston mapped this entire pathway out using a tiny microscopic roundworm called C. elegans.
Oh, right, the worm.
This specific worm is famous in biology because every adult has exactly 959 cells. No more, no less.
That is so precise.
It really is. Every single time this worm develops from an embryo, the exact same specific cells are genetically programmed to die at the exact same moment to shape its body. They found the literal genes that control the execution.
Which is wild to think about. And you, listening to this right now, you actually benefited from this exact programmed death.
Oh, absolutely.
When you were an embryo in the womb, your hands looked like solid paddles. You had webbed fingers.
Yes, you did it.
The only reason you have individual distinct fingers right now is because the cells making up the webbing between your digits executed their apoptosis program flawlessly, shrank down, and vanished without triggering any massive inflammatory reaction.
It is an essential part of daily life. We eliminate tens of billions of cells every single day through apoptosis just to maintain our intestinal lining, regulate our immune system, and ensure our organs don't grow infinitely large.
So apoptosis sounds like the perfect, elegant system, but what happens when that meticulous programming breaks down?
This raises an important question, and it shifts us into what is arguably modern medicine's biggest ongoing battleground. We intuitively think of disease as cells dying when they shouldn't, like a heart attack or a stroke.
Right.
But sometimes the most dangerous disease is a failure to die. Cell death isn't always bad. In fact, failing to die when commanded to is the foundational pathology behind cancer.
Because a cancer cell is essentially just a severely damaged cell that actively refuses to self-destruct.
Exactly. Healthy cells detect their own DNA damage and trigger apoptosis. But tumor cells frequently accumulate genetic mutations that specifically disable their apoptosis pathway.
It's like having a quality control inspector on a factory assembly line. Normally, if a defective product comes down the line, the inspector hits the big red emergency stop button and trashes it.
Right.
But in cancer, the inspector has fallen asleep at the switch, so the defective, damaged products just keep rolling off the line, dividing endlessly.
That is a perfect analogy.
Yeah.
And the most famous inspector in human biology is the p53 tumor suppressor gene. It is mutated or disabled in roughly half of all human cancers.
Half. Wow.
Yeah. The cell is severely mutated and should undergo apoptosis, but the emergency stop button is broken, so it survives and divides uncontrollably.
So modern oncology, a lot of it is basically trying to force these rogue cells to remember how to die.
Right. Much of traditional chemotherapy works by causing such overwhelming catastrophic DNA damage to the cancer cells that it forcefully overrides the broken switch and triggers apoptosis anyway.
So it forces the issue.
Exactly. And newer, highly targeted drugs, like venetoclax, work by directly attacking the specific anti-apoptotic proteins that cancer cells build to block their own death. We are biochemically forcing the tidy death program to resume.
But there's a flip side to this balance, too. If cancer is a disease of too little apoptosis, what happens if there's too much tidy death?
If the program becomes chronically overactive, it leads to devastating tissue loss over time. Excessive apoptosis is deeply implicated in neurodegenerative diseases like Alzheimer's and Parkinson's.
So the brain cells are dying when they shouldn't.
Right. Neurons in the brain that should have survived a long, healthy life are improperly receiving signals to execute their self-destruct programs.
It's a razor-thin balance. But, there's another major misconception in the sources that I want to make sure we hit before we wrap up.
Oh, what's that?
We've been talking about apoptosis as this quiet, clean, tidy process. Leaves peacefully falling from a tree. But the literature points out that not all genetically programmed cell death is tidy.
No, it's not. And this is a relatively recent major paradigm shift in molecular biology. There is a specific programmed pathway called pyroptosis.
Pyroptosis. Pyro meaning fire.
Exactly. It is
Yeah.
a genetically encoded deliberate death, just like apoptosis, but its purpose is the exact opposite. It's triggered by specialized intracellular infection sensors called inflammasomes.
Okay.
When a cell realizes it has been invaded by a virus or a bacteria that it cannot defeat, it doesn't just quietly fade away and let the virus replicate. It deliberately, violently explodes itself.
It literally sets itself on fire to warn the neighbors.
It swells, ruptures its membrane, and intentionally releases massively potent alarm signals, things like the highly inflammatory cytokine IL-1 beta, to rapidly recruit immune cells to the area to fight the infection.
Which sounds incredibly heroic, like the cell jumping on a grenade. But this is actually what made the COVID-19 pandemic so incredibly deadly for so many people.
Yes, unfortunately. This inflammasome-driven pyroptosis was a major driver of the infamous cytokine storms we saw in severe COVID-19 patients and what we see daily in hospital sepsis.
So it's an overreaction.
Exactly. The virus wasn't necessarily just killing the lung cells directly. It was triggering this highly inflammatory, programmed cellular explosion on such a massive systemic scale that the resulting immune response caused severe tissue damage and multi-organ failure.
Oh, wow.
Yeah, a programmed, orderly-sounding biological process produced some of the most chaotic, destructive inflammation imaginable.
So what does this all mean? When we zoom out and look at everything from the spreading wavefront of a heart attack to the precise zone 3 destruction of a Tylenol overdose to the vanished webbing between an embryo's fingers, every major medical reality we face is fundamentally dictated by this microscopic machinery.
It really is.
Whether it's a leaky boat filling with calcium that crosses the irreversible point of no return, or a mutated cancer cell where the quality control inspector fell asleep, our overall health is entirely dependent on our individual cells managing their own destruction.
It completely reframes how you look at human biology.
Yeah.
We spend so much of our conscious lives obsessed with how to stay alive. We diet, we exercise, we avoid physical danger.
Yeah.
But at the cellular level, the ultimate goal of a truly healthy cell isn't to live forever.
No.
No. It's to know exactly when and exactly how to die for the good of the whole.
That is just a stunning way to think about the body. But I want to leave you, the listener, with one final thought to mull over today that builds on all this.
Okay.
We talked about cells dying messily, dying tidily, or refusing to die and becoming cancer. But there's a fourth, deeply weird path we're just starting to understand. Cellular senescence.
How?
Sometimes a severely damaged cell refuses to undergo apoptosis, but it also permanently stops dividing. It just sits there, suspended in a twilight state, secreting low-level inflammatory signals. Scientists call them zombie cells.
Which is a very fitting name.
Right, and researchers now believe the gradual accumulation of these immortal, malfunctioning zombie cells in our tissues isn't just a side effect of getting older, it might be the literal biological root cause of human aging itself.
It's a fascinating frontier in pathology.
Thank you for joining us on this deep dive. Next time you read about a new medical breakthrough or hear an ambulance rushing down the street, we hope you remember the microscopic ticking clock, the silent wavefront, and the incredibly complex machinery operating inside every single one of your cells. We will see you next time.